Hi, I'm the postdoc of the University of Southern California. So in my last vlog, I have introduced the background of the single-cell sequencing and the T-cell development. And in this vlog, I will follow the study to introduce how they use the single-cell sequencing to interpret the biological event during the T-cell development. And if you want to completely understand this vlog, I will recommend you to watch my last vlog at first. And I will put the link on the bottom of the video so you can click the link to watch the last video.
Hello everyone, today I'm gonna to present a paper from Cell Journal. This paper's name is Single-Cell Sequencing Reviews Regulatory Gene Expression Dynamics, Leading to Lineage Commitment in Early T-Cell Development. So this is the logic flow of the central questions and the breakdown of the specific technologies this study used to answer their question. And I will follow this flow chart to digest this study for you. And firstly, the question they raised is whether the dynamic ATP are heterogeneous. They use the in vitro cell culture technology to answer these questions. Firstly, they use 3 markers to isolate the cells from the thymus, which is the KIT, CD44, and CD25. And the cell they try to isolate is KIT positive, CD44 positive, and CD25 negative cells. After that, they culture the cell into the 96 well plate and observe their development under the microscope every day for 6 days. And they are knocking the marker of the DN2 cells, which is the BCL11B, into the cells. So if the cell expresses these markers, then they can observe the signal in the cells, and which is labeled by the blue fluorescent. So they culture the cell for 6 days with the T-cell induction medium, and after 6 days, they observe a lot of cells expressing the BCL11B and also the CD25, which is another marker for the uncommitted DN2A cells. And according to the observation, they found almost all clones generated cells expressing CD25 and the BCL11B by day 6. Only 2 of the 66 columns produce more non-T lineage cells resembling granulocyte. Therefore, according to this result, there are over 90% of viable chronogenic ATPs processed T lineage precursor activities. And then they use the bulk RNA sequencing measurement to profile the RNA transcriptome of ATP and the DN2A cells. And here is the transcriptome expression pattern of the ATP and the DN2 populations. It's easily to find the difference between the ATP and the DN2A populations. And we can find the EPT expressed non-T genes, such as this group, indicate that there's a subpopulation of EPT may go into non-T cell lineage. But there's a little bit difference between these 2 populations. And these populations are similar to each other even though they express different level of the ALT3. And they also express the T lineage markers, GATA3 and the TCF7. This gives us the conclusion that even though there are some T lineage markers expressed here, most of the cells are already contribute to the T cell lineage fate. And here are 2 questions. The first question is that how many substates were comprised in ETPs? And the second question is that how homogeneous cells progressed through ETPs? Here the answer to this question is that after they try to isolate the so-called ETPs, they find there a little bit population may go to the granulocyte but not a T cell lineage. And they also use the bulk RNA sequencing to identify the ETP from the DN2. So the next step is that they're trying to answer whether the stem or progenitor and the T cell genes are co-expressed. So they use the single-cell seqFISH and the C1-smartseq to identify the putative non-T outgroups or markers, and also trying to find the co-expression pattern of the stem and the T cell genes. So firstly, they trying to use the seqFISH to identify the co-expression pattern of the T cell genes and the stem cell genes. And here is the workflow of this technology. The cells are firstly immobilized on the cover slip for imaging-based profiling, and after that, they use the HCR seqFISH to detect the 55 low and medium copy number RNA in the individual cells, and use the HCR-smartseq to detect the left 11 high copy number RNA. And after that, they use the immunostaining to confirm the RNA expression is consistent with the protein expression. And they compare these two different RNA single-cell sequencing, and they find only the seqFISH can detect the low copy number RNA, such as the Notch1 and the TCA7. But these two markers are very important for the T cell lineage establishment and maturation. So that means even the transcription factor is very important. Given to their low copy number in the cells, they are very hard to be detected by the normal classical RNA sequencing or single cell sequencing technology. But the seqFISH can solve this problem. And then they use the immunostaining to confirm the RNA expression is consistent with the protein expression. And here is the answer to a question that raised before, because a lot of people just challenge RNA expression pattern may not reflect the expression pattern of the protein, which is the executor of the function of the cells. But here, they show the protein expression pattern and the RNA expression pattern is consistent in the single cell sequencing. So the first line of the chart is the RNA, the transcript expression level, and the second line is the protein expression pattern in the cells. And when they compare the expression level of these two different grades, most of the RNA sequencing data can reveal the expression pattern of both RNA and the protein level. So next, they analyze the seqFISH data by correlate different cluster of the genes. And the pairwise co-expression pattern show two distinct groups, which is the T association group and the stemless group. The stemless group is here, and the T associated group is here. And if we look into this, and we can find the non-T or progenitor groups is totally different from the DN3 genes or T-associated genes because they have non-association value. And two groups of the cells labeled T or innate lymphoid cells, which is here and here, containing members of GATA3 and the Icaros family. They are correlated with both the stemless and the T-associated groups, which means there may be the transition step or substate. And the stemless groups also correlated with the non-T group, which is here and here. So this is a very interesting result that indicate that maybe within the stemless group, some cells are contribute to the non-T cells. So here I want to give an explanation of how to look into the heat map here. So each column represent for one individual cells, and each row represent one genes. And if we look into this heat map, we can find the cells have been already clustered. And how they do this? So they use the non-markers to label the cells and then generate the heat map and the t-SNE analysis. And then they can create the trajectory of the cells that take during the development. So after they use the non-markers to establish the landmark and supervise the clustering the cells, 9 clusters are found based on the endpoint genes, starting from the earliest ATP cells to the committed DN3 cells. And the progenitor genes were not all co-expressed, but instead displayed distinct although overlapping patterns. So how do we interpret this sentence? Here is the progenitor cells here, and we know that some cells are very progenitor genes, such as the MLT3 that were expressed in a more restricted developmental pattern, many at cluster 1 and 2. And the KIT and SPI1 is also stem and progenitor genes, but they are expressed until stage 3 and 5. So that means some genes are revoked earlier, but some genes will express there until the T-associated genes appear. The GATA3 and the TCF7 were activated at substate 3 and 5, where the FLT and LMO2 have gone. So here is what we have said before. Some genes will be there until the GATA3 and the TCF7 appear, but the FLT3 will disappear earlier than this gene appear. So only the cluster 8 was low in TCF7 expression, which is here. We can find this cluster of the cell expressed totally different from any other previous cluster. So that's the question, that whether this group of the cell are non-T lineage. So to answer this question, they use another deep sequencing single-cell technology to confirm the supervised clustering cells. The advantages of this technology is that they can detect a lot of transcriptome, but the trade-off is that they can only detect a low cell number, and they use two different analyses to automatically cluster the cell. The first one is the PCA, we generally use in the microarray. And according to the PCA analysis result, the cell can be automatically clustered into 4 groups, according to the marker FLT3 and the BCL11B. But if we use the t-SNE, which is more commonly used in the single-cell sequencing, then the cell can be clustered into 10 clusters, which is more separately distributed among the map. And if we map those non-markers back to the t-SNE analysis, and we can find some markers are restrictedly expressed in a specific group of the cell, such as the BCL11, which is distributed in the angle of upper right, and the FLT3 is restrictedly expressed in the bottom left angle. So that means this group of the cell may be the more mature DN2 cell, and this group of cell may be the progenitor of the T lineage. And consistent with the seqFISH data, the cell can be automatically clustered into 10 clusters. And again, separating the ETP and the DN2 cells, because the first group of the cell here express the most of the stem and progenitor genes, and the end cluster, the cluster 7, only expressed the mature T cell markers. So these two groups of the cells are very distinct from each other. And here is another cluster which is similar to the cluster reviewed by the seqFISH before, which is the cluster 9. So here is the cluster that raised the possibility again, maybe they're the out group of the cell. And here comes to another two interesting gene, which is the IRF8 and the MPO genes, because they are highly correlated with the TCF7 but not the BCL11B. We know that the TCF7 appeared earlier during the T cell establishment, but not the mature marker of the T cell lineage. So why these two genes that's supposed to be non-T cell marker are highly correlated with the early cell marker of the T cells? And also the multipotency associated genes such as SPI1, FLT3, MO2, MAF2C, CD7 were all frequently co-expressed with the TCF7 and GATA3 in individual ETPs. So that raised the question, maybe all of the genes are expressed transiently during the transition stage. So according to this and this, we know that there are a lot of markers that appear at different stages of the T cell lineage transition and maybe indicate the substate of the T cell development.